All right, let's get started. So today our topic is about high throughput sequencing and read mapping. And so there's the outline for today. We want to very quickly go over different sequencing technologies. The first generation sequencing is Sanger sequencing, and Sanger actually won the Nobel Prize for inventing this technology. The idea is you need to have enough amount of the same DNA, many copies of the exact same DNA, and we only sequence one strand at a time because the other strand is just a reverse complement of the first strand. And if you have multiple copies of the same DNA in single-stranded, then initially this was done in 4 different lanes. So in each of the tube, you add DNTP, which is ACGT, everything, plus a DD and so for example, in the first tube is ACGT, everything plus a DD ATP. And so basically, with the one strand in there as a template, polymerase will try to make the other strand of the DNA by incorporating, in this case, C, a G, and A. But at each time it incorporates an A, there is a chance of incorporating the correct A, but also the DD ATP. In this case, once it's DD ATP, it cannot continue anymore. And so there will be some molecules that end at this location because it's an A. Some molecule will end the next time there's an A because whenever the DD ATP gets incorporated, it cannot extend anymore. And so this will happen on the first tube. In the second tube, it's all of ACGT plus the DD-CTP. So anytime you stop at a C, if it's incorporating a DD-CTP, it cannot continue to extend. Then there will be a number of molecules stopping at this point, and so on. And so these 4 tubes of reactions are run separately in the machine. And then the end result of this tube with a mixture of DNA will be run on a gel. And depending on where this molecule ended, you can see that they stack up. For example, the shortest molecule in this case is the C, is the first that it runs furthest. And then the next one is going to be a T, and the third one is going to be an A, and so on. And so basically at the end, when we look at a specific gel picture, we can actually read it out if the furthest band is an A, so we all know this is an A, second base is a C, next base is a T, next base is a G, and so on. And so basically, you sequence one long molecule or one type of sequence at a time, and you run this in the 4 tubes. Later on, there are ways to kind of merge the 4 tubes together. So actually, the DD NDP, the NTP are given different colors. So they're on the same gel, but then have different colors that you can read out directly. But this is the first generation sequencing. You sequence one kind of DNA at a time. Second generation sequencing really is the dominant technology right now, and the market leader is a company called Illumina. And what they do is something called a massively parallel sequencing. Basically, you have a tube of mixture of DNA. It's whatever DNA you have, and you attach the adapters to the 2 ends. And then there is a flow cell, which is kind of a glass slide. And you pour your DNA on top of this glass slide, and the adapter will be attached to the bottom because there are some probes on the glass slide. And then in each location, there is an in situ PCR amplification. And so you can see they kind of try to make the DNA bend over and use the other adapter. And so because the 2 ends have 2 different adapters, you can then using this kind of little bridge to amplify everything in between. And after they are amplified, you dissociate. And you can see with different cycles, so they bridge over, create a template, amplify, and then you open them up again. And with a few cycles, basically in this spot, there will be multiple DNA molecules having the identical sequence around here. And that's how they generate, this is called cluster generation. So basically, you pour your mixture of DNA onto a glass slide, and each molecule in that location will try to make a few copies of itself. And once that's in place, we can do the sequencing. And this is sequencing by synthesis. So the idea is, remember on this glass, or kind of a glass slide, each location will have one type of molecule with the same sequence, but there are actually multiple copies of it. Here we're only showing, for example, supposedly this is one type of sequence, this is another different sequence, this is a third different sequence. Each of them we're only showing one molecule. The reality is in that location, there's a cluster of molecules with the same sequence. They are in situ amplified. And so basically with this in place, you flash this glass slide with just one nucleotide, or sorry, in this case, ACGT, 4 nucleotides at a time. And each of them is only allowed to attach once. So basically, you cannot add 2 nucleotides at the same time. Basically, in order for the nucleotides to be attached, they all have to look like this, right? But if the DNA, so they grab onto the next one, they grab onto the next one, they grab onto the next one. That's how the DNA chain can really continue. But if the nucleotide you flash doesn't have this handle, then they can add to the previous one, but they cannot continue. So every time you only add 1 nucleotide at a time. So in this case, you can see this particular location depends on the template. A C is being added here. That's different DNA molecule in here. G is being attached here, and another molecule, another DNA sequence, a T is being attached in here. And then at this time, you hybridize and you wash it off and then shine lights on it. And because the C, G, T have different fluorescent color, in that location, you will see a different color showing up. And so you will know, okay, this location just incorporated a C, this location incorporated a G, and this location incorporated a T, and so on. And then after the imaging, the dye is cleaved, and also the termination groups. Basically, now the molecule is allowed, so you can kind of give it something that the next molecule can be added now. And then you repeat this cycle. Now you flash the ACGT again, and each of the previous template can now add 1 more nucleotide into this location. And then you hybridize, you wash, you use imaging to shine lights again and see which molecule is being incorporated in which location. And then this cycle just repeats. And so nowadays for a sequencing machine, whether you sequence 50 base pair, 100 base pair, or 200 base pair, roughly the time is proportional to the whole run. The length of the run is proportional to how many bases you want to sequence. So if you want to sequence 150 base pair, the runtime will be about 3 times longer than another sequence run with only 50 base pair, okay? And so if we look at, so there's some YouTube videos to show this. If this is an example of, this is the flow cell. It's like a glass slide. You can see many, many different colors in here. So this is a zoom in of this location. You can see many little dots, and the different color indicates different A or C or G or T being incorporated. And so you can imagine this is the original template DNA in this location. It's not just one molecule, it's multiple molecules with the same sequence. And then when you try to add a T in here, you can see here is a terminator. With this in place, the next molecule cannot be added. So you only add 1 nucleotide at a time. And when you shine lights to it, this location will give out the T color. And so in the first time in this, you can see here, if we zoom in from this to now look at this, within this location, this spot in the first cycle, you have a kind of a blue fluorescent color showing up in here. And the second cycle, a green fluorescent color showing up, and that's G. And in the third cycle, a orange fluorescent color gets incorporated, that's a C. And in the fourth cycle, a T gets incorporated, 5th is A incorporated, and so on. So you can imagine in this reaction, at every cycle, you shoot a very high resolution image of this whole flow cell, which has millions of, by this time, hundreds of millions of spots. And it's a huge image, and every cycle, you get a very high resolution image. And at the end, as long as you line up the image together, you will know at which cycle which nucleotide gets added. And so with this, we can sequence hundreds of millions of DNA sequence on this one flow cell at a time. And that's why it's called a massively parallel sequencing. And this is totally different from the first generation, where we sequence one at a time, another one at a time. Right? And so right now, the market dominating company is Illumina, and they have a fleet of sequencers from the very small, kind of baby type that you put in your lab to the really big ones that like NovaSeq that usually only sequencing facilities or companies can own. But for example, this NovaSeq we showed on Tuesday, the capability is that you can generate about 6 terabytes of data per run, which is about 40 billion reads, and it's paired and 250 base pair in length, right? And so it's really quite amazing. You can see here the price of genome sequencing is decreasing very much, but recently it's kind of stabilized a little bit. And it's a possibility that new technologies are still to be developed, but also Illumina currently really does not have competition. I think with competition, they might even decrease this more. So the third generation sequencing is really single molecule sequencing. And you don't even need to amplify DNA anymore. Right, so in the first generation sequencing, you do need to amplify DNA, and it's 1 tube per molecule. You amplify this many times. Second generation sequencing, you don't need to separate them in tubes to amplify. You can pour them onto this glass slide and amplify in situ many, many molecules together. Third generation sequencing is really single molecule. Basically, you use either polymerase or a pore where the sequencing is going through the pore and giving out different signal depending on whether it's A, C, G, T being incorporated or going through the pore. And this, you don't really need amplification. So far, there are some technologies such as PacBio or Oxford Nanopore, and they use like slightly different technologies. You can see each of them kind of needs a kind of a polymerase type. This is a polymerase. This is kind of just cleave the nucleotide and going through the pore. You can watch the videos after this. These companies have released some preliminary, like prototype machines or reagent. They usually generate much fewer reads, probably in the order of thousands or tens of thousands of reads, whereas Illumina can generate 40 billion reads in one run, right? That's the difference. But their reads can be much, much longer. You can sequence a 10 kb molecule in one sequence, whereas Illumina right now is usually 100, 150-ish, right? So depending on the application, if you want to say now there's a new virus, you just want to sequence from the beginning to the end, maybe the third generation sequencing would be better. But if you need something that needs a lot of counts, you need to know the quantification levels, then second generation sequencing is better. So that's the overview of sequencing technologies. Any questions on this? Okay. So as a computational biologist, you just need to know roughly all the sequences that are coming in, and you have to deal with the data. So now we are going to teach you how to look at the data. The raw data coming from the sequencing machine, thank God, is not a big image or many, many layers of image. What they will give you is already a sequencing file. This Illumina company has the software to do. And so basically what we get is a FASTQ file. And for each sequence, you'll get 4 lines. The first line is the ID of the sequence. The second line is the actual sequence. The third line is, you can see they have a different preface, right? If it's an @ sign versus a + sign. @ sign is the sequence name, and the + sign means that this is the quality of, the line for the quality of this sequence. And then you see some other nucleotide, like, oh, this doesn't look like a nucleotide. There's some other characters in here, you can see here. So every sequence has 4 lines. two IDs and the actual sequence and also this quality score. The quality score follows the first generation sequencing convention. They use ASCII code to decide the quality by 33 different characters. And this is basically, you know, on the ASCII, how the small number is a bad quality, the higher number is good quality. And so basically, based on this, you can see which location has better quality than others, and decide whether some read are so bad that you don't want to use it. And the Phred quality, this ASCII number roughly represents the negative 10 log 10, the probability that this base pair is read incorrectly. You know, what's the likelihood that it is wrong? And so if this whole number, you know, because we did a minus 10 log 10 of this probability, the bigger the number, the better the quality. And so why do we need to do quality control? So because the sequencer output is just a read, if with the quality score, you get this FASTQ file. You want to kind of know whether my sequence data is okay. What can you do if your the data quality is not so good? Yes, there's a question. I guess like I would think it would like get worse if it not really. Yes, it is the case that as you go, the sequence quality would be worse. I'm not sure this is a real one. We just kind of randomly generated something for you, for example. Yes.
Would you mind like what the letters thing, like the letters N stand for? What's the real score for N or for V for X?
Is on the quality score part. Yeah. Oh, basically these are just characters. So you can imagine if the quality score is only 0 to 10, you can just use numbers to represent them, right? But if you have 33, then obviously you will need some characters as well. And in this case, they even added the different characters. So basically, it's just this is in the actual encoding of these characters. This is a small number and this is a higher number.
Yeah, but is there any, like for example, I think that A stands for 11. I think he's asking for the ASCII value.
Yeah, so beneath them, each of them has an ASCII value.
So we just need some like sheet of table.
Oh, good thing is you don't even need to do that. All of this is provided to you. I'll show you later. Yes, another question.
I guess, like, what's the range of the incorrect probability? Like, is worst quality goes from like 99% wrong, and best quality like 10 to the minus 33 or something?
Yeah, something like that. Okay. Right, something like that. Yeah, so another question.
Does that mean like the best quality would be like a lowercase 0, z?
Yeah, it will be a lot of on the other side. But really 10 to the minus 33, it's like a ridiculously small number. And so you can see most of the time, even if you're here, it's pretty good already. Okay. So what can I do if the sequence quality is not so good? You have to decide whether this is just one read or even a single nucleotide. So you can imagine, as we mentioned, when you sequence, what people see is that usually the very first couple, a few, maybe 3, 4 nucleotides, they are not so good because kind of machine is getting in the groove to start sequencing. So initially there might not be as good data and you can just trim them out from the beginning. And then after that, it's usually very good quality data and then it just go off over time. But occasionally maybe some base pair might be bad, just one base is bad. Those are actually okay. If the overall the read is good, you can still use it. But you know, like if it's just one nucleotide is okay. If the whole flow cell, all the other reads are okay, but just one read is bad, sometimes it's because 2 molecules were too close to each other. And when they are amplified, every time these spots are giving out 2 different colors, and you're not sure what's going on with these 2, right? So that you can just ignore that spot. That's also okay. If you have just one sample that's bad, because right now with Illumina, in each lane of sequencing, you can already put many, many samples in there. If only this sample is bad and all the other samples in that lane is good, it just means the person who has done the experiment preparing the sample for sequencing needs to go prepare their samples again. They didn't prepare it correctly or something, right? For example, there are some experiments you want to sequence something, but you don't have enough starting material, trying to scrape off like DNA or RNA from one cell or something that you couldn't amplify. And then you put it on the sequencing, that sample doesn't give you a good result. That's the sample preparation step. If the whole run is bad, meaning that all the samples, all the lanes are giving you bad quality data, it means that the sequencing facility should resequence it for you for free, right? So that's why you kind of need to know this. The difficulty here is the FASTQ files are really massive, right? So there needs to be an automated way to do quality control. And for that, there is FastQC or FastQ or quality control. And this is kind of interesting. This code has been used by almost every one of us who do sequencing analysis, except that this has never been published as a paper. It's just a kind of a cool tool that we all use. So bioinformaticians created a good resource for the community without even a paper. And so FastQC has different levels of quality control. For example, if you look at your sample that might have 5 million or 50 million reads, and you look at the quality of the reads from the first base to the second base and so on, you might notice that in the first nucleotide, the quality is pretty good. You can see here it could be 38 or 32. So I would say usually about 30, these are pretty good quality reads. They all can be used. So here they are cutting actually by 28, and even above 20 is pretty good. And so if, and then you can see the quality will decrease. This is the average of all the reads at this location, right? This is and so as you go further into the reads, you can see the quality start to decrease, and this kind of decrease at around 50 base pair. But I think for this one, even if they were to sequence for another maybe 20 bases, it will probably be okay, right? Because these are all still good quality data. Whereas the bad quality data, even at nucleotide 20, things are falling down, and like at 25, you are not getting really good results. And so sometimes even if you can read a 250 base pair, after the QC, you might want to only look at the first 200 or maybe first 150, right? So this is a good way to look at this. So the good quality is consistent and also high quality along the read. Sorry, there's a question.
How does Illumina calculate the probability
So the question is, how does Illumina calculate this from imaging? As I mentioned, for example, if the spot is very, very big and it's very, very clear, there's only one color, there's no different color overlapping with each other, they calculate this. I actually don't know the detailed algorithm, how they did it. They probably tried a lot of different tests to calibrate their read quality. But this basically now, when you run the machine, by the time you get the data, it's already calculated. Okay, other questions? Another good readout is if you look at so the FastQC has a different pages of output. Another output is actually similar to this. It's derived from this figure. You are looking at for each sequence, you can look at, because each sequence is very long, they have many, many nucleotides. Every nucleotide has a quality score. What is the median quality score along the read? Right, so because if the most of the read has quality of 38 or 39, you will probably get a median score close to 30 something. So I would say anything with a median score above 30 or 20, even 28 is a very good quality result. But then sometimes you might see this, a whole bunch of reads for some reason, their median quality score is only around 16 or something. Well, it's just lower. It will give you some sense, maybe this is not a good quality sample. Another interesting thing is to look at the nucleotide content on each location. As we mentioned, this has been noticed by people. Sometimes I'm actually showing an extreme case. This situation usually doesn't happen for that long, occasionally you might see the sequencing machine in the first couple cycles, you might have very unbalanced GC or the AC-GT balance. It was kind of a bit crazy. Then it's okay. When you see this, it's actually a pretty good quality run. The most ideal case is actually it's all 4 lines together. It means you have loaded the dies completely even, right? They are exactly the same. But even if they are a little bit off like this, as long as they're fairly parallel, this is pretty good. Sometimes when you see something like this, you can decide to trim off, in this case, probably the first 15 base pair. You say, oh, somehow the sequencing machine here is kind of a bit crazy. Let's just remove the first 15 base pair and only map the remaining one. But sometimes when you have a sample like this, it would mean that in one cycle, all the reads you see is an A. In another cycle, all the reads you see is a G, and so on. There is something wrong with this run completely. We would probably need to rerun this whole run. Okay, maybe they just didn't add the things correctly in the different cycles. This is not very common. And finally, is to look at the sequence GC content. We believe this should roughly follow a kind of a normal distribution. And if you look at all the reads, what is there, on each read, because it's like 100 base pair long or so, what is the GC percentage? It should be pretty, close to, in fact, because human genome, a lot of genomes are less GC rich, so you get maybe 45, that's pretty good. But if you have something like this, you might want to check the quality a little bit. So I would say most of the time, we just get this. The average quality, if you get something like 30, that's good enough. Okay. So questions about our FastQC? Next, we deal with a sequence alignment or sequence mapping. The idea is that once you get the sequence, what you have, you don't really care what it is. It could be a human sequence, it could be a mouse or some pollution from the air, bacteria, virus, whatever, DNA sequence, it can be sequenced. And so in the very first step, after we check the sequencing quality to be okay, the next step is to map the sequence back to the genome that you intend to do. Supposedly, you are growing some human cancer cells in a petri dish, and you're treating the cell with a drug. You isolated the RNA, and you convert that into a cDNA, then you can sequence. After you get the layer of sequencing data for your sample, you probably want to map that to the human genome. In fact, if you map it to the mouse genome, you might find that only 3% of your read can find a location in the mouse genome. But if you map it to the human genome, you might see maybe 80% or 90% of the reads mappable to the human genome. Okay, so that's a read mapping step. So there are a number of algorithms we want to show you. The first is, read mapping in general is, supposedly this is human genome, 23 pairs of chromosomes, and you now have one query sequence, the new sequence you just got from the sequencing machine. You want to see, this read is coming from chromosome 1 location this, right? You map it to this location in the genome. There has been algorithm done. The big challenge here is mapping hundreds of millions of reads back to the reference genome. Not only takes a lot of CPU, but also takes a lot of memory, and it's memory intensive and slow. And also what is even more challenging is that a lot of the times, even if it's a human cell, as we know, you and me probably, our DNA can occasionally differ by 0.1%, but it happens. And sequencing machines also sometimes make a mistake. As you can see from the quality database, maybe they're not so confident. And so a lot of the read mappers also allow some mismatches in the first 30 base pair. As you go later, there could be even more mismatches because of sequencing errors. The beginning, usually you don't see so many sequencing errors, but you still can allow some mismatches because you can imagine if we only allow mapping of 28 nucleotide out of 30, 4 to the 28th is a really humongous number. You can still uniquely identify a certain nucleotide sequence in the genome, right? So even if you have two mismatch, yeah, you still should know where it comes from in genome size like human. But if you allow for mismatches, it will be much slower. And sometimes there might be small insertions and deletions. These are called indels. If you miss one nucleotide or add one nucleotide, those indels will even further slow down your read mapping algorithms. The very early days when we are still doing first generation sequencing, people use algorithms like these type of seeding type of algorithms. For example, supposedly you have a genome, let's just say human genome is this, and you can build an index for example of k-mers, and this is a k-mer of 5, all the 5 mers, right? So the first 5 mers, CGTGC, CGTGC, and you can say, okay, this appeared at location 0, but this is computer scientists, right? They start from 0, and and then this is a position 4, and then the next one is GTGCG, that's here, and so on. And you can see this, it's all the things are kind of ranked in here. Everything starting with C, or actually everything starting with A should appear on the top, and things at the bottom should, with T will appear on the bottom. But you basically build a very big index. And then, yeah, when you have a query sequence, the idea is, this is an algorithm that is available at NCBI. It's one of the most widely cited algorithms, because in the early days, anybody who sequenced something, they do a blast to see whether this is a new sequence or not. And so it's blasted against all of the other sequences in NCBI. And the idea was, if you have a new sequence coming in the query, you also divide the query into k-mers. If your index previously is a 5-mer, well, in reality, it's more like a 20-mer or something. And if your query sequence coming in, you also break it up into k-mers. And then for each k-mer, you look up this table, you find, okay, where is the match? Very quickly you find the match. And then you try to extend that initial match a little bit further and say, can I do a little better than this, right? Yeah, you find all the database k-mers that match well with it, and you select some cutoff to say anything that match at least with a cutoff are kept in there. And then with the initial seed of the matching, you try to extend until, okay, they don't match anymore. They form something called a high scoring segment pairs, which means that on this one long sequence, there's a region that match here, there's a region match, there's region match. And the next part is we want to only keep the statistically significant high scoring matches based on, in this whole database, existing database, what's the likelihood you will see some kind of a match like this. And after you have these significant scoring matches, you ask, is there a database sequence? Supposedly A is your database sum sequence and B is your query sequence. You see, they actually have a number of these high scoring pairs. And can I try to kind of stitch them together and then generate the final alignment? And the final alignment is using something called Smith-Waterman algorithm. When I started my bioinformatics education, that's how I started. But for today's, because we are not using it as much, you can watch the videos on YouTube from previous lectures. Questions?
So this effectively handles copy number.
This one hasn't really considered the human genome copy yet. This is just, if you have any kind of a database of sequence, how do you align this? In fact, nobody is using this approach nowadays to align this to the human genome. It's way too slow. If you have one sequence, it might be okay. But this is not what we're going to show you in terms of the current Illumina sequencing read mapping. This is what we learned in the 1980s and 1990s, okay? Yeah, so basically you find some initial seed match and try to extend them, and then you find, oh, this sequence and in a query and my sequence have a number of these high scoring pairs. You try to align them together, then you get a good match. Okay, that's kind of the very early generation sequence analysis. The next thing is suffix array. This is basically, well, so first we need to show what is a suffix tree. And so suppose that you have a text. In this case, we're showing, like say an English book with something called like banana. You can build a suffix tree which contain all the substrings in this sequence. So for example, that it will have a number of these suffix, B-A-N, first is banana, is anana, nana, and Anna and Na and A, right? So that can build into this tree. And it has been used by some alignment tools in the early days. The nice thing, so you can see here, there's the tree. If you want to look at the whole banana, it can go directly here. But you can also go if you want to go, say Anna, it's, A and NA and NA, right? And then that's position 1. Yeah, so you can see banana directly here is 0. Na is position 1. And nana is, you start with N, A, and another NA is position 2. And so for this tree, it takes order N to build the tree. N is the size of the genome or your database of sequences that you try to match. But it takes order of M time to search, and M is the length of your sequence, your query sequence, how long you want to do this. Unfortunately, if you have a genome or database that's really big, this tree is huge. It can be, human genome can be 50 gigabyte. And so people also use something called a suffix array. And so the ith entry on this array corresponds to the ith smallest suffix in this whole tree. And so you can see here, the smallest one is a $sign, and it's a $, and all the A's are together, and the ones that is bigger is kind of later in this suffix array, and then it's B and N and so on. We will show you in the next lecture. Actually, this approach has been used for RNA sequence matching, RNA sequencing. It is implemented actually quite well, and it can run very fast. It still takes time, order N, to build this suffix array, but it still takes it takes a M log N to do the search, and the index is kind of moderate because you just need to store this information. And if you have a lot of sequences that are kind of similar, they can share cells in here. And so basically, in order to search this, you go to the middle of the sequence. Go to the middle of the index and ask, my first nucleotide, if it's A or above, okay, you are on the right track. But if it's bigger than A, you take the half of this, and you basically every time you go to half of this to find the location, then you will find the index. Depending on the implementation, this actually is a good algorithm, but in DNA sequence, it hasn't really taken the dominance. It's actually being used in RNA sequence. In DNA sequence, we want to show you another algorithm. And the algorithm that's really used a lot in DNA sequence alignment is using Burrows-Waller transformation. And these are called BW aligners. And if you search on Google Scholar for Burrows-Waller aligners, there are 2 major contenders. BWA was developed by Sanger Institute, and you can look at these citations. These 2, there's like first generation, a slightly improved version there. And then the second was Bowtie, which was developed at University of Maryland, and then this group now moved to Johns Hopkins, and it's a Bowtie family. And you can also see here, so both algorithms have been cited by over 20,000 times, and it's really used a lot. They actually use very, very similar idea and just slightly different implementation, and they roughly came out around the same time. And so in order to really understand how this is done, we need to first teach you Borel-Waller transformation. This is a computer science technique in order to do text compression. Yeah, so supposedly you have a database of sequence. You can imagine this could be very, very long. We are only showing you a very short sequence. And at the end of the sequence, we add a $sign. Every time you end the sequence, you add a $sign. And so in order to do the Borel-Waller transformation, you do something which is kind of interesting. Every time you move the last nucleotide and put them in the beginning. So for example, original database is this sequence, right? We move the $sign to the beginning. And so now it is $sign A, C, A, A, C, G. In the next one, you move the G to the beginning, and everything else is the same. And the third one, you move the C to the beginning, and everything else follows, and so on. So this part so far is pretty easy, right? It sounds like a pretty simple thing. In the second part, what you do is you rank the sequence. After you have this in place, you rank all the sequences based on their nucleotides, because $sign is the smallest, is on the top, and you can see AA appears before AC, and ACA appears before ACG, you know, and A appears before C, and the CA is before CG, and so on. So you rank all of the rows. This actually is gonna take some time to do, but you only need to do it once. And once you have ranked all of the sequences, the last row is, or sorry, the last column is what you need to record. And that is your BWT transformed thing. And this is the only information you need to store, which is kind of weird. It almost looks like the same as your original sequence, except that now the order is changed. And you're wondering why we need to do this. So once the BWT tree is built, you discard everything. All of the remaining ones are left, are discarded. You only keep the very last column of information, right? And the way people use it is because for compression, you can see here, when you align them together and this together, you see here, there are 3 A's right next to each other. Imagine you are trying to compress a whole big book together. There will be many cases you have the or similar words together. So there will be many words that have, you know, maybe T in here, H in there, E in the next one, right? And then here is T, H, E, T, H, E, and so on. And so those will all be consecutive rows, right? And so what happens is that during the compression, you can say, okay, this is G and C and $sign. And instead of writing A, or in the THE situation, write many, many times the same thing, you can just say, this is followed by 3A. That's how the compression came around. And so the computer scientist who developed this was really using this for text compression, but then the computational biologists in the class or who learned this technology thought, oh, maybe we can use it for sequence as well. You can actually use it to convert a human genome sequence into this BWT. But why do we need to store this sequence? Is because once you have this sequence like this, you can completely recreate the original sequence using something called LF mapping. So I will show you how this is done. So in LF mapping, remember we only record this last row in here, sorry, last column in here. And this is the original database sequence, right? And so what we do is, if you see this A in here, it's the second A in the last column. This is equivalent to the second A in the first column. That's the magic property of Boras-Waller matrix. Once you sort them, the second A in the last column is the same location as the second A in the first column. And because this second A is in this location, and the last character in here is a $, it actually tells us this is the beginning of the sequence. This is position 1 of the original sequence, right? So I will show you, if we have the this last column, how we construct the total original sequence. It's done like this. Remember, this is the last column after we have sorted everything, right? We sorted everything based on from the beginning of the sequence, but now we keep track of the last column. Because we have a G, we know that the beginning is a $, which is the end of the sequence, right? So we know that the sequence ended with G, because the $, would be added to the end. And because this is the first G in the last column, do you know what is this sequence corresponding to? It corresponds to the first G in the first column. And so from this location, you know that it's the same sequence as the first G in the first column. And then we ask, okay, if this is the first position, what is the last position on here? And that's a C. And we will know that the previous nucleotide here, or the previous sequence in here, is a C. Remember, the way we construct this Boras is because we always move the last nucleotide and put it to the front, right? And therefore, if you see G as the first nucleotide and C as the last nucleotide, you would know that this C actually appeared before the G. Does that make sense? And so, and from this C, because this C is the second C in the last column, see there's another C before, and this is the second C in the last column, we will know that it is equivalent to the second C in the first column. And then because we go to this row and we ask what is the nucleotide before that, it's an A. So we'll know, okay, the previous one is A. Right? So you'll be able to write the previous letter. And because this is the third A in the last column, this is equivalent to the third A in the first column. And from this first, and then we look at this row, because the last letter is an A, we will know that before this A, the previous letter is also A. And because this is the first A in the last column, we will know that it's equivalent to the first A in the first column. And then look at the previous nucleotide, which is at the end, which is a C. And from here, we will know that this is the first C, therefore we'll go to the first C. And then we have an A, the previous letter is A. And this is the second A. Let's see, this is the second A in here, which is similar to, which is the same as the second A in the first column. And now we see a $, and we know we have finished. That's how you write out the full sequence. Yes, question.
This is done with your query sequence?
No, this is, we haven't started looking at the query yet. This is just how do you look at the whole database of sequence, like a human genome, right? So we need to, basically you convert the original human genome sequence into this matrix, and you write out all the letters out, you sort them, you create this matrix, and you can have a compression of this. Question?
Where's the $?
The dollar sign just tell you have reached the end of a chromosome, or you have reached the end of the whole database. Okay, so you know that you shouldn't keep looking anymore. That's the end. It will tell you no more matches. Another question.
Sorry, you just said that we only need the last column and we can discard everything else. But it seems like here we need both the first and the last column.
The first column is just the dollar. Yeah, actually, you just need to record how many dollar signs, how many A's, how many C's, how many G's. That's it. Does that make sense? That you can construct from the last column. Okay, now I get it. Okay. Okay, so now if you have the whole matrix, the question is, I have a query now. I have this database. I have this database already in this index. How can I find the correct alignment? So supposedly my database is very long, and after the transformation is already in this form, and my query is very short. I'm just making it very short. AAC, can I find the location of this sequence? And for this, we also use this LF mapping approach. It's done like this. So initially, we have a sequence C. We just look at this whole, the first column. Remember, the first column now is just, you just count how many dollar signs, how many A's, how many C's, how many G's, right? And how many T's. And so you can very easily find that first C in here. And the last C, you have a range. You have a pointer for the range. But then you ask, if the previous nucleotide is an A, that should appear at the very end of this alignment, right? So if you see a C and an A, how many are in that range? You look at the end. So for example, the ones that has C probably is this much, but if you have to look at the end, also have to, the previous nucleotide has to be an A, you need to know what is that range. It will be a smaller, it's usually a smaller range, but in this case, because it's so short that we're getting the same thing. And from this range, you ask, okay, these two are at least matching to the A. What is this A? It's the second A on the last column in this case, which means that it will be the second A on the first column in here, right? And then here, this pointer is the third A on the last column. This should correspond to the third A on the first column. Does that make sense? Therefore, you go next to the, you move your pointer now, because anything in between this range, so far it still matches, because they do have a C right after, and they have an A before this, right? So everything in between this pointer still map on the first two characters. And then you ask, okay, if we look at here at the end, where do we still have an A previously? And this one is not a match because it's dollar sign. It means that it has reached the end of the sequence, so that's not a match. This one is an AC, and the previous nucleotide is an A. It still is a match. And so basically now our pointer, it was from here, and then afterwards, the dollar sign is removed, and A is still a match. And you look at this A is on the it's the first A on the last position, which is equivalent to this A, right? You go to there, and there you end with the pointer. And so because the sequence ends here, you will know that this is a match. But very often in the database, well, sometimes in the database, if you keep matching, you can imagine initially your pointer is very long. You have all the possibilities. Every time you look at the back, you shrink it a little bit. And that will move to a portion like this. And then it will shrink a little bit, right? And then it will move to, and as you go, the pointer, the top pointer and the bottom pointer will be closer and closer and closer to each other. And eventually, if you have finished matching all your query sequences, it means that you have found a match. Anything between the pointer, you know that these are all matches of this in your database. But if at the end your pointer closed and you have nothing left, that tells you you don't have a match, right? That there's no match in the sequence. So yeah, basically in progressive rounds, we have a top pointer and a bottom pointer, and it delimits the range that still the query sequence is still matched. And you look at the previous sequence, you look at the previous nucleotide, you look at the previous nucleotide until you finish mapping everything. And so the mapping is from the end to the beginning until you finish the whole query. If the range becomes empty, you finish everything, the top and bottom pointer now has collapsed, then it means that you do not have a match. In that situation, if you want an exact match, you can just say, sorry, there's no match. However, what you can do is you backtrack. You say, okay, what if we allow one mismatch? The previous nucleotide, we can change one. Or the previous 2 base nucleotide, we can change one. And previous 3rd base, we can change. You can increase your range a little bit. But you can imagine that part of the implementation is much, much more complicated. And that's how Bowtie and BWA can each be used by 20,000 people, right? Cited so many times. And so the backtrack basically allows for mismatches. And also the backtrack implementation can allow for indels, but those will be much, much slower. The direct match is actually pretty fast. You just look at the query, because actually for this database, in addition to keeping track of the last BWT made this sequence, in the very first, we actually, because you can imagine a 3 billion base pair genome is very, very long. What you can keep track is, I have X number of $signs, then I have X number of A's, X number of C's, X number of G's. You can keep track of this so you can go to the location very quickly, right? And so this way, if you know, okay, this is this, so you can imagine, you can say, oh, this is a 40 million A in that genome. You can very quickly go to that row, right? Because you keep track of this. And so that can help you figure out whether you have a match. The question then is, if you know you have a match, where does this match come from in the genome? That you also need to come up with very quickly, right? Not only you need to know my query sequence matched 3 locations in the human genome. You also want to know this location is chromosome 1 at this place. That's next one is chromosome 3 at this location. And that is done by, initially it's by backtracking. You can see here, once we have found the match, you can see this is a perfect match, AAC in here. We look at the last character, which is a C. This is the first C on the last column. It's equivalent to which C is the first C in the first column, right? And so from the first C in the first column, you ask, what's the previous character? It's an A. And that's the second A in the last column. This is equivalent to this one, which is the second A in the first column. And then at this point, you see the $sign. You say, that's the end of the sequence. Therefore, how many times did we backtrack? This is 1 step, 2 step, 3 step. We reach the end of the sequence. Or at least we know the original sequence must be this whole thing. Then we know, this is location 3, right? So in some sense, you can backtrack all the way through. You will know where that is, but you would be like, that sounds pretty dumb, right? If you have to do all the backtrack, it's gonna take too much time. So in reality, in the first column, not only we keep track of how many A's and C and G and T's, at alternating at certain rows, you also say, for example, you say this row is really chromosome 1 some location. And then a few rows below, you say, okay, this one is really chromosome 3 some location. Therefore, hopefully, after a few backtrack, you don't need to go all the way to the beginning of the chromosome. You already hit a row that has an index. You can stop there. And that will give you the location of the sequence. A question?
Yeah, 2 quick questions. One is, what's the average compression that you get on?
So in reality, for human genome sequencing, read alignment, we don't do compression at all. They just use it to speed up the search. They don't actually compress the sequence. Yeah, so but index is still manageable now with 3 billion sequence. Oh.
Second quick question. Is that the third index from the right or the index?
This is third index on the left. On the left. Yeah, so, but basically, in theory, you have to do backtracking all the way to figure out where your match is. But if you can just keep track of some of these rows, where they come from, it can help you speed up. And after a few maybe a couple backtrack, you'll hit a row that has an index, then you can know where your sequence is. Okay, it's the, yeah, so how many steps did you reach this location? It's the few nucleotide after that has the hit. Okay, so that's how, yeah, so basically, we can store the index of subsets of these rows in the first row. And sometimes you even store the last column a little bit, because you can imagine the last column is also, it's kind of, this one is not in any particular order. So sometimes you have to keep track of, how many A's in there, just remember, okay, where are the A's and C's and G's and T's, so that in order to get to the first C or the whatever 50 million C, you can get there much faster. Okay, and so yeah, so basically this is the summary of the Burrows-Wheeler alignment. Basically, once you have a genome you want to map your sequence to, you build this Burrows-Wheeler transform matrix to store the entire reference genome as a lookup index. And then once you have the index, when you have the FASTQ sequence coming in, you align the reads base by base from the end to the front, from the end to the front. And each of them will find the alignment. And all the active locations are reported. For example, after you finish mapping, if supposedly the pointer has just one row left, that means this sequence is mapped to a single location into the genome. But imagine you do the sequence and then there's a range, it would mean that it's mapped to multiple locations into the genome. And the algorithm will report all the locations that it's mapped to. And if there's no match found, you can do a backup or try a substitution, backtrack, which will then be much slower. But these are parameters you can tune how much mismatch do you allow and things like that. The general idea is like this. The implementation difference on how the backtrack is done, how often do you have these indexes, these are what makes difference between BWA and Bowtie, but really both of them use the similar idea here. And so basically what you have in order to run a program like this is you need to have the algorithm, which is either Bowtie or BWA. You need to have your input sequence, which is in a FASTQ format, your query sequence. But you also have to have a database, your human genome, that's already built into a index form, which has the BW last column information, but also the index of some of these rows that keep track of the address. Okay, so that's that you can just directly download from the web. And we already installed them on the Odyssey server, so you can use it. And with that, you can align the sequence. So Ben Langmid actually have video. So he is the developer for the Bowtie algorithm. You can also watch his video about how to do the BW transformation and how to do the RF mapping and how to do the alignment. Okay, questions? After we finish the sequence alignment, what you will get is output from this. And at the time, several genome sequencing centers decided a standard. They call it a SAM file. It's alignment file. And if you look at the header, it looks something like this. The very beginning is, you have information like chromosome, length, how many chromosomes do you look at, and the genome assembly. For example, it has information, human genome by this time has different versions of annotation. The newest is NCBI 38 genome, HG38. In this genome, we have however number of chromosomes, each chromosome, how long it is. So that information is in the header file. And also, it tells you the sequencing platform. This is in the early days, the Solid, which no longer exists, this machine. But they're also, Illumina is the current sequencing platform. And there's also read group. Read group is useful in some cases. Supposedly, people put their sample in order to make sure that from run to run or lane to lane, there's no bias. Sometimes people want to spread their DNA sequence to be run on multiple lanes. If there is GC bias in one, it doesn't interfere with the GC bias in another lane. So at the end, all the DNA sequence for the one sample needs to be concatenated together. And the read group information is still kept because the normalization is done read group by read group. And after that, you can normalize. This information might be useful, but you may or may not need to even worry if you have all the read in one lane. And this will also tell you, in this case, you are running a BWA and at what version in order to get the final sequence alignment. So in all the SAM files at the very top, you will get this. You will know kind of what machine sequence is, what program do you use to do the alignment, what version of the human genome are you trying to align to, and how many chromosomes are there in this genome, and so on. So those information, and there's some other comments. But after that, this is the actual mapping result. So basically, this is the read name. You can see this is the read name. And then this is the mapping. Yeah, so if it's 0, it means it mapped to, it is okay, it's mapped to a particular location. If it's 4, it means we could not find this location in the genome, which is not rare. If you sequence some human samples, it's very, very common for about 10 to 30% of your sequences to not map to a human genome. It could have some contamination, some germs in the air, and so on. So 4 is unmappable. 16 means it is mapped, but it's mapped to the reverse strand in here. Okay, and next is the, so next is the actual mapped location. This sequence is mapped to reverse strand, chromosome 9, this particular location. This is the actual sequence that you get, and this is the sequence quality score. So actually, SAM file contains the information from your original FASTQ file, because this is the read name. This is whether it's mapped, where it is mapped, and this is the sequence, and this is the quality score. And then, so this one, in case you have some mismatches on that sequence, it will tell you where that sequence is in the mismatch. So for example, this one, everything matched, you have no mismatch. Yeah, NM is number of mismatches, this is 0. But in this one, you can see here, there are actually 2 mismatches of this sequence back to the reference genome. But where are they? You can actually read it in here. The first 3 is a good match, but in this location, the reference sequence is a C, but in our sequence 1, 2, 3, 4, it's an A. So you know that the reference genome is a C, but our sequence is an A. You can read from here. And then we have another 30 nucleotide that's matches. And then in the reference sequence is T, but in our sequence is 1, 2, 3, it's this base. Our sequence is a G. So they tell you where the mismatch appears, and then there are 3 perfect match, right? And you can see here, this one is unmappable, so we didn't find a location of this guy, right? So you can see this, okay? And so at the end, you will just get this SAM file. You can visualize this directly. Oh, by the way, the SAM file can also be compressed into a binary file, and these are called BAMs. In the homework, later on, you will see that in alignment and actually nowadays, all the computational algorithms that deal with data, they can deal with SAM files. But if you want to see what the SAM files look like, you can still output a SAM just to visualize it. You will see that it has a header and it's just like rows and rows and rows of this kind of thing. Every sequence is aligned. It will tell you whether it finds a location, whether it finds multiple locations, or it didn't find a place. Okay, but you can compress it into a BAM file, which contains pretty much all the information in your original FASTQ that you can go back to. And algorithms can deal with this to look at the count, look at differential abundance, but you can also just visualize this on a genome browser. In the labs, you will see some genome browser in here. And this is an example of reads. These are actually cDNA reads that are mapped to the exons. You can see the reads are on this exon, the reads are mapped to this exon, and you can see the reads in here. But the introns, there are not so many reads. So each of these little block here, it indicates a read is mapped to that location. Okay. There's another file format called the BED file, which people don't really use to look at the full alignment. It's usually used to store genomic intervals. Later on, you will see in the ChIP-seq lecture, you can use it to represent peaks in the genome, or you can annotate genes in the genome, certain interesting locations. So it's an easier format in here. It just says it's chromosome 1, starting position, ending position, and you can give it a like peak 1, peak 2, like you can give it a name, and you have some scores and whether it appears in plus or minus strand. You can use it to represent your read alignment, but people don't do it. In the very early days, people tried it, but you can see it loses a lot of information. You just know some coordinate, but you lose some information. And then this is the BED file specification. And this BED file can also be compressed into something called the big BED. It's just a binary form that is a smaller form that people can do computations on very quickly. But generally, for most of the read mapping, you will get a BAM file, okay? And so that's the summary for today. So there are first generation sequencing, second and third, and most of the time in this course, we're gonna give you second generation sequencing from Illumina. And the sequences you get from this sequencing machine is the FASTQ file with 4 lines per sequence. And then you first use FASTQ-C to do quality assessment of your sequencing. And then you will use algorithm to map the read back to the genome you are interested in. There are different ways to do this. There's a BLAST approach, which is really slow, but people don't do this for high throughput sequencing. STAR use suffix array, which we will see in the next lecture, how we do RNA-seq mapping. And BW and Bowtie both use Boros-Waller transformation and LF mapping to find the location. And finally, the aligned reads are stored in the SAM or BAM files. So in homework 1, we're going to ask people to try to do some alignment. You just we don't allow do too much, 1 million reads. You can run it and see, what the input file, what's the output file. Okay, great. Thanks.